teman-teman serta seminar yang telah sabar menunggu. Okay, so we have now 29 participants joining in the rooms. Okay, hope I can recognize some of them. Anyway, okay. All right, Rhesi, please play the video. So we have something to hear and to see, to watch, okay?
With over 17,000 islands, Indonesia is often called the world's largest archipelago. All these islands cover only about 30% of the whole country. The other 70% is water. This in mind, it is easy to see how immensely diverse aquatic ecosystems develop here. Indonesia is a country with great potential in marine mega biodiversity. Qualified human resources are needed to research and get a better understanding of this extraordinary potential. This will have positive impacts on many levels for future generations. Proper infrastructure and developing knowledge are the base to be able to go deeper into modern marine research. MERO, which stands for Marine Education and Research Organization, is supporting this specific line of research. MERO was founded in 2020. This institution is located close to the sea on the famous Raya Secrets muck dive site in the Tulamben area. The Tulamben area is home to a wide range of various underwater habitats, including a shipwreck, coral reefs, sand slopes, rocky areas, as well as muddy inflows from rivers. MERO students have the possibility to explore biodiversity directly by diving here. MERO's laboratory is well equipped with the latest in technology, where not only marine research is done by a qualified team, but a special aim is to offer the possibility to students from Indonesia to learn and share on a national and international level. Already collaborations are active with many national and international scientists and students. MERO's laboratory provides research equipment for microbiology, morphology, chemistry, DNA processing, as well as a scanning electron microscope. It offers students and researchers accommodations like dormitories and private rooms. There is a spacious classroom for courses and seminars. More buildings like an additional laboratory, a restaurant, and a showroom are planned. MERO was built in the idea of sustainability and eco-friendliness in mind. Electricity is provided by solar panels. Wastewater gardens take care of the sewage. Garbage is separated, and the organic parts directly composted on the grounds. Being in a rather rural area, MERO also supports the local community with education in regard to garbage management, and there is a Sunday class for the children from the village. The very heart of MERO is certainly marine education and research in general, and the research of nudibranchs in specific in collaboration with scientists from all over the world while offering students to take part in this process and knowledge. MERO is ready to welcome interested students and researchers.
Hey, all right. That's all the video. So from Xu-Wen. So it was a last year video, so we don't have the new one yet, but there are certainly a lot of updates and facilities are new in the facility in the MERO labs. So as you might aware that the labs, the foundation is located in Bali, which is famous for tourist destination. So at the end, you are coming to Bali, for example, you are on holiday and at the same time doing research, or conversely, you are doing research and holiday at the same time. So it's quite nice positioning. Anyway, all right. So okay, everyone. So Rhesi, shall I hand it to you for the for the webinar, or?
Yes, I think we can start now, Pak Wayan.
Okay, all right then.
For the opening, yeah.
Okay, all right, perfect. I let you to handle the rooms, the rest of the conference. Thank you.
Yes. Okay. We will start. Good afternoon, ladies and gentlemen, wherever you are, maybe in China, Japan, and then also in Indonesia. So, Assalamualaikum warahmatullahi wabarakatuh. Om Swastiastu. Salam sejahtera, namo buddhaya. Salam kebajikan for all of us. So let me introduce myself. My name is Rhesi Kristiana. I'm a chairman of MERO Foundation, and also I will be a moderator today with Prof. Xu-Wen. First of all, I would like to say thank you for the present of the Honorable Dr. Rahmadi Prasetyo as a founder of MERO Foundation, Iwayan Mudianta PhD as a board member of MERO Foundation, and also Professor Xu-Wen as a speaker today. Thank you very much for your willing time to us, to the Indonesian student and the researcher, for your knowledge and sharing to us. Okay, today is the third webinar that held by MERO Foundation in 2022. The first webinar is about MERO Foundation Information Day, and then second, the webinar is about marine chemical ecology and secondary metabolite, and also marine microbial symbiont. And now today, we will learn about marine natural product potential drug leads. So now, let me share the screen a little bit. Yeah, this is today is about bioprospecting of marine natural products by Professor Xu-Wen, and I would like to go to the next slide about the participant. So today there are 122 participants that already joined. Mostly they are 64% student, and then lecturer, researcher, and then undergraduate, and also the other institution. So this is the institution from the many national university in Indonesia, from west to the east of Indonesia, and also from several research institute, National Research and Innovation Agency Indonesia, and also from pharmaceutical. So we would like to welcome to all of you, the participant. Thank you for your time today. So I hope you can enjoy the webinar today. And next, this is the rundown of the webinar today. We'll be opening, which is now, and then next the opening from the Dr. Rahmadi Prasetyo. He will deliver about MERO Foundation, and then a little bit video tour lab, and then we will go to the introduction of keynote speaker, and then presentation by Prof. Xu-Wen, and then discussion, and the last is documentation. So MERO Foundation arranged several series in the 2022 to provide the communication media for capacity building, program update, and also science update for the student, specialist, researcher, and also government, so they can have a room to discussion, to have a good discussion about marine. As we know, Indonesia has a huge diversity of the area and also marine organism. So there are so many potential that still undiscovered. So please, participant, enjoy the time today and take a note if you have any question later about Professor Xu-Wen Materia. Okay, so now we go to the opening by Dr. Rahmadi Prasetyo to open this webinar today. So I welcome you to come, Pak Dr. Rahmadi Prasetyo. Are you already there, Pak Pras? Halo, Pak Pras.
Ya, halo, ya.
Yes. Okay, we can hear your voice, Pak Pras.
Ya, terima kasih. Tes suara, apakah suara saya bisa didengar ya?
Yes, clear, Pak Pras.
Clear ya? Ya.
Yes.
Assalamualaikum warahmatullahi wabarakatuh. Selamat siang, salam sejahtera bagi kita semua. Ya, pertama-tama kita bersyukur di pasca pandemi ini kita masih bisa diberi kesehatan dan kekuatan untuk bisa terus bersinergi, berjuang, menuntut ilmu dengan webinar pada siang ini. Pertama-tama saya ucapkan terima kasih kepada teman-teman dari MERO yang sudah mempersiapkan acara webinar ini, dan juga untuk Prof Xu-Wen yang boleh hadir pada webinar kali ini. Thank you for Professor Xu-Wen for your time to share with us about the bioprospecting of marine natural products this afternoon. Dan juga pada siang ini kita bersyukur sekali ya, MERO sudah ketiga kalinya dalam tahun ini kita bisa melakukan webinar ini. Ya, sebelumnya ada suara di belakang saya, mohon maaf, saya masih ada di bandara. Nah, saya masih di bandara Cengkareng ini. Oke. Ya, next. Ya, next. Ya, ini MERO adalah satu institusi riset ya yang sama-sama kita ketahui bahwa kita sama-sama membangun ini untuk baik untuk kepentingan riset maupun edukasi seperti itu. Kita berharap ada banyak sekali mahasiswa maupun para dosen yang boleh join dan bergabung dengan kita. MERO berada di Tulamben, ya sekitar 100 km dari Denpasar. Next. Kita berdiri, ya silakan next. Kita berdiri baru sekitar 2 tahun ya. Lokasinya ada di pinggir pantai. Ini adalah keanekaragaman yang ada di MERO, cukup baik. Dia langsung berada kalau di sebelah utara langsung ke laut, dan sebelah sisi selatannya adalah Gunung Agung yang sangat cantik. Jadi suasana sangat mendukung juga untuk melakukan riset dan memikirkan hal-hal yang terbaik tentang teknologi kita ini. Next. Ya, tujuan daripada MERO ini ya dalam jangka panjang maupun jangka pendek adalah untuk mensupport pembelajaran tentang local people di sini, juga menyediakan sarana dan prasarana bagi para mahasiswa maupun dosen maupun Bapak Ibu para peneliti ya, baik swasta maupun negeri, untuk bersama-sama mengembangkan potensi kelautan kita. Ya, sama seperti saat ini kita sekarang sedang lakukan adalah bagaimana kita belajar sampai ke negeri Cina ya, belajar dari profesor dari negeri Cina untuk bagaimana memanfaatkan bahan-bahan yang sangat prospektif dari marine natural products seperti ini. Ya, next. Ya, kita selama sekian tahun ini ya, sejak tahun 2020 sampai 2021 ini ada beberapa hal yang sudah kita lakukan. Ya, kita sudah melakukan banyak hal. Di awal kita di tahun 2000 kita sedang menyelesaikan beberapa konstruksi, khususnya beberapa lab, juga pengadaan peralatan-peralatan seperti itu. Kemudian kita membangun jejaring dengan banyak pihak, banyak institusi. Dan saat itu juga kita mulai juga menerima beberapa mahasiswa maupun dosen yang sedang riset hingga saat ini. Ya, next. Next. Ya, ini adalah beberapa fasilitas yang ada di MERO. Kita ada kamera area for morphology, kemudian ada lab tentang microbiology, kemudian ada DNA extraction and PCR, ada chemical room, kita juga memiliki scanning electron microscope atau SEM, kemudian fungi room sedang kita set up, dan juga NGS sedang kita set up. Ya, kita berharap peralatan ini cukup untuk mendukung berbagai riset-riset dari teman-teman, para mahasiswa, rekan-rekan dosen untuk mengembangkan penelitian tentang kelautan di Indonesia ini. Ya, next. Ini adalah beberapa aktivitas-aktivitas dari berbagai mahasiswa dari seluruh Indonesia dan juga dari dosen, baik tentang morphological observations, microbiology, terus juga ada SEM. Ya, ini SEM observation ini salah satu peralatan yang ya kita sungguh bersyukur ya yang kita miliki bisa mendukung banyak teman-teman untuk melakukan riset ya. Mudah-mudahan ini juga nanti dengan kehadiran NGS juga semakin melengkapi kita. Juga ada extraction proses yang ada di sana. Oke, next. Ya, teman-teman semua dan juga para peserta, ini ada beberapa current research yang saat ini sedang MERO sedang lakukan atau kerjakan. Ya, barangkali ini ada ide-ide ya dari teman-teman semua, Bapak Ibu dan rekan mahasiswa yang memiliki topik serupa, boleh join juga ya. Saat ini kita juga di tahun 2022 ini ada Torrey Grant yang sedang kita lakukan, ada Joint Usage Research Center on Tropical Disease General Joint Research ini juga ada, tentang nudie brain survey juga kita sedang lakukan. Ya, ini beberapa kolaborasi juga sudah kita lakukan, baik dengan Undip maupun dengan Stifar, IPB, dan beberapa kampus-kampus yang lain. Sudah mulai bergabung, berkolaborasi dengan kita semua. Next. Nah, tadi yang seperti yang saya bilang, SEM Results ini adalah seperti ini, suatu peralatan yang sangat-sangat mendukung untuk kita melakukan observasi secara detail ya, baik itu tentang radula dari nudibranch sudah sangat terlihat dengan sangat jelas di sana. Kemudian nanopartikel silika pun yang punya size 900 nanometer juga bisa terlihat, dan juga sponge juga sangat jelas di sana. Next. Ini juga database kita tentang bakteri dan Actinomycetes. Ya, silakan next. Ya, teman-teman yang lain silakan bila ada yang punya koleksi atau interest tentang beberapa marine invertebrate, ya kita juga memiliki beberapa sampel collections. Barangkali bisa join bersama-sama dengan kita. Ya, next. Ya, beberapa mahasiswa saat ini juga sedang melakukan internship maupun final projects, baik itu S1 maupun S2 ya, dari berbagai universitas. Saat ini yang sedang berjalan adalah dari Brawijaya, kemudian dari Udayana, dari Gajah Mada, dan Sriwijaya, juga dari Undiksa. Mereka sedang melakukan berbagai topik yang ada di MERO, dan juga beberapa mahasiswa sedang lakukan juga dari Diponegoro University, kemudian Hasanuddin University, maupun dari Brawijaya University. Ya, next. Harapannya kita di dalam waktu mendatang ini, kita seperti sekarang kita mulai ya. Jadi dengan adanya pemanfaatan prospek dari marine natural products ya, dari farmasi seperti itu ya. Makanya kita hadirkan salah satu guru terbaik ya dari negeri Cina. Ya, kita hadirkan Profesor Xu-Wen ini orang yang luar biasa yang bisa membekali kita barangkali menginspirasi kita untuk mengembangkan berbagai produk marine natural produk yang lain. Ya, next. Ya, kita juga ada di waktu mendatang juga sedang mengembangkan juga tentang marine conservation. Ya, kemudian juga ada plastic waste processing seperti itu. Mikroplastik seperti itu ya. Juga kita berharap juga di dalam MERO ini kita juga berkolaborasi ya dengan local people ya, masyarakat lokal untuk kebersihan pantai ya. Seperti itu kita juga mendapatkan grant dari International Coastal Clean Up seperti itu. Ya, kita berharap media ini atau sarana ini bisa memperkuat para peneliti untuk berjejaring, berkolaborasi, bekerja sama dengan masyarakat sekitar ya, dengan bersih-bersih pantai, dengan juga kita membersihkan lingkungan sekitar kita, juga kita bersih-bersih di laut juga. Underwater clean up juga kita lakukan, river clean up juga kita lakukan. Selain juga beach clean up, dan itu kita lakukan pada hari Minggu, jadi kita sebut sebagai Sunday class. Ya, next. Kita juga mendapatkan grant dari tim SIS ya, dari Ocean Conservancy USA. Kita juga melakukan beberapa kegiatan seperti beach clean up, ada underwater clean up, juga ada river clean up. Ada aktivitas kita juga kita support school round of MERO Foundation ya. Kita berharap memang di dalam MERO ini kita membangun kolaborasi dengan anak-anak sekolah ya. Kita juga sedang lakukan dengan SMA, SMP seperti itu supaya mereka punya wawasan tentang kelautan sejak dari awal. Kita juga bekerjasama dengan US M.Si ya, dengan berbagai aktivitas di sana. Juga kita lakukan plastic exchange dengan masyarakat lokal ya. Dengan masyarakat lokal ada plastic exchange. Ya, kita berharap juga kita bisa berkontribusi ya untuk lingkungan laut kita khususnya. Next. Ya, teman-teman, Bapak, Ibu, dan rekan-rekan semua, itulah sedikit kilas balik, kilas tentang MERO ya. Silahkan dicatat, silakan di print shoot juga bisa ini. Kontak detail kita baik di berbagai media ya, baik email maupun di website maupun di Facebook, Instagram. Kita berharap kita bisa bekerja sama, berkolaborasi, membangun jejaring, bersama-sama kita membangun kesempatan bersama untuk mengembangkan diri menjadi pusat studi kelautan di seluruh Indonesia ini yang berpusat di Tulamben ini. Hingga terima kasih, begitu aja mungkin ya Bu Rhesi. Mudah-mudahan kita semua mendapatkan manfaat pada siang ini, kita semua diberkahi, dan dengan demikian maka webinar pada siang ini saya buka dengan resmi. Silakan. Terima kasih atas waktunya. Wassalamualaikum warahmatullahi wabarakatuh.
Terima kasih banyak Pak Pras. Thank you very much Pak Pras for the explanation of the activity in MERO Foundation. So this is very thoughtful and also open our mind about how the importance of the marine potential and the future of the world. Okay, because several participants also just joined. So next we will play the video again of MERO Foundation before we continue to the Materia. So please the video tour of MERO Foundation.
Welcome to Laboratory of MERO Foundation. First, I want to show you the camera room. In this room, we're designed for sample handling and visualize morphological sample, and also we sterilize lab equipment in here. So, here we have autoclave. This is binocular microscope and stereo microscope. And this is magnetic stirrer with hot plate. And for your information, our laboratory is really close to the sea. And then we will move to the next room. So this area is a more open area to prepare blood activities. And in this side we have chemical area to extract the bioactive compound. So we have multi-centrifuge. We have Xu-Wen. We have separator funnel. This is rotary evaporator. This is fuel hood. This is freezer -20 degree. And in here, we have deep freezer for -80 degrees Celsius. And we will move to the microbiology area. This area were used to culture microorganisms such as bacteria and actinomycetes. So this is bacteria fridge. And in this side, we have molecular area. So we have multi-vortex, centrifuge, heating blocks, and this is laminar airflow for DNA extraction. So in this side, we have media fridge. We have colonical blood. This is incubator. And this side, we have analytical balance and vortex. We also have laminar airflow and biological safety cabinet class 2 for works with human pathogens such as multi-drug resistance. And we will move to the next one. So this is a molecular area for PCR room. So in here we have thermocycler. This is qubit and fluorometer and microsphere. This is gel electrophoresis, and this is gel visualization. And we will move to the last one. So here we are in the last row. In this row, we have low vacuum scanning electron microscope. We also have smart filter, so we can process non-productive sample, such as biological sample. That's all our laboratory facility. We also have other facilities such as dormitory, classroom, private room, office, and restaurant. And we're looking forward to host researchers from around the world. Thank you.
Thank you, Dila, for the video. Okay, so next we will continue to Prof. Xu-Wen, but before that, let me to read the rules during the session. So it will be in Bahasa, Prof. Xu-Wen, so please await for a while. I will read this for the participant. Yeah, so yeah, this webinar, it's not only by Zoom. We also put the live streaming in the YouTube. So if another participant cannot join in Zoom, they can go to the YouTube. Jadi I will read in bahasa. Demi kestabilan koneksi internet, video dan audio peserta akan dimatikan secara otomatis dan akan dinyalakan kembali saat sesi diskusi berlangsung. Pada sesi diskusi, gunakan fitur Q&A pada Zoom untuk mengajukan pertanyaan. Bagi yang ingin menyampaikan secara langsung, dapat menggunakan fitur Raise Hand pada Zoom. Bagi peserta yang hadir di YouTube, dapat menyampaikan pertanyaan melalui live chat. Dan kemudian link absen akan diedarkan melalui menu chat pada Zoom dan live chat pada YouTube ketika acara berlangsung. Ya, Okay, so this is all the rule for the participant. So next, we will go to directly to Prof. Xu-Wen Lee. Before that, let me introduce Prof. Xu-Wen Lee. Prof. Xu-Wen Lee is from Shanghai Institute of Materia Medica Chinese Academy of Science. In 2022 until present, he's working in Shanghai Institute and then research group leader and researcher, and also in Bohai Rim Advanced Research Institute for Drug Discovery, Yantai, China. And then in 2014 to 2015, Prof. Xu-Wen Lee also doing his postdoc in Zurich, Switzerland. And then the PhD was done in University of Paris in French. And then for master in China about natural product chemistry. And then for bachelor is in Tianjin University about pharmaceutical engineering. His research interest focus on the chemoecology guided discovery of diverse and complex marine natural products, biomimetic synthesis, and function-oriented synthesis of bioactive MNP toward new marine drug leads. And this is, he also has so many publications in the very good reputation international journal. So this is your time to hear Prof. Xu-Wen Lee about the knowledge, and then please take a note and discussion and ask as many as you can to know more about pharmaceutical product. Yeah, so especially in Indonesia, the government also support us to how to release the result of research to go to the society, so it's become such as drug or benefit for food. Kalau di bahasa Indonesia, nenek moyang belajarlah sampai di negeri Cina ya. Yeah, so Prof. Xu-Wen Lee, we also in Indonesia we have a little statement. It's if you want to learn, you have to learn until you go to China. Yeah, so we are so happy to welcome you now. So now the time is for you, Prof. Xu-Wen Lee. Yes, yeah, please. Still mute, I think, Prof. Xu-Wen Lee.
Okay, so can you hear me now?
Yes, we can hear you now, and now the participant is almost 100. Yeah.
Okay. Thanks a lot, Kristiana, for this kind introduction. So I will show my my slides now. Can you see my slides?
Yes.
Yes. Okay.
Yes, very clear.
Okay. Thank you. Good afternoon, everyone. Thanks a lot for MERO Foundation for this kind invitation to do a presentation about the marine drug discovery based on marine natural products. So my title would be like the Blue Ocean Drug Treasury. I would like to guide you for a journey of marine drug discovery. Okay. So for the start, as we know that life originated in the ocean, and from the simplest single-celled creatures to the longest plants, algae, and the biggest animals, blue whales. So the marine have the most colorful life species, animals, plants, and microbes. So that would be my opening. Okay, just want to attract all you guys with these beautiful pictures. Okay. So as we know that marine biological resources mainly contains plants, animals, and microbes. And among them, animals are the most diverse marine species. As you will see that there are cute animals like these very beautiful cute mollusks, and there are also ugly ones, And there are also giant ones and the very beautiful, colorful ones. And also in the deep sea, there are a lot of fluorescent marine animals. Okay. And of course, the marine biological resources have a lot of applications. So the first is food and food additives. You see like a lot of like seafood. I think that you guys will like very much. And also the pesticides and biologics, such as these. This is the horseshoe crab. The blue blood of the horseshoe crab can use for the detection of bacterial endotoxin. Okay, and also for health products, there are a lot of applications by like the deep sea fish oil and a lot of products that made by the marine biological resources. And also cosmetics. I think the young ladies will like these two brands very much, La Mer and La Prairie, which is very like a luxury cosmetic brands. Okay, so some of the products come from the marine biological resources. And of course, the last but not least important is drugs. So this is the drug is the highest value utilization of the marine biological resources. So like the anti-cancer drugs, the anti-Alzheimer drugs, and antivirus drugs. So there are a lot of drugs that are directly or inspired by the marine natural products, which originate from the marine living organisms. So in terms of the drugs, in fact, back to the ancient China, there are already the records of marine biological resources as the traditional Chinese medicine. So back to the 11th century BC, a book which called the Book of Songs already have like 18 marine medicinal organisms for the drug for use as the traditional Chinese medicine. And afterwards, there are a lot of other medicine books in China has records of marine traditional Chinese medicine. And back until 2009, the book Chinese Marine Materia Medica has recorded over 2,000 marine medicinal biological species for the TCM. So this is TCM for the modern marine drug discovery. It becomes from the 1950s. So basically, it is the marine drugs are derived from the diverse metabolites, and the metabolites is originated from the rich marine lives. They have to adapt to the special marine conditions, because we know that the marine conditions we have like high pressure, high salt, and low oxygen, and there are a lot of complex interspecies relationship. So the marine lives, they have to live in such conditions with producing diverse and functional metabolites, secondary metabolites, to be their chemical weapons or do their chemical defense. So these metabolites would have some medicinal functions. So the explore of the medicinal function of these metabolites could be a rational utilization of the marine biological resources. So up to now, the marine lives for medicinal development is less than 1%, but there are already like 40,000 of marine molecules, marine natural products has been discovered, and there are about 20 drugs has been approved on the market, and there are still over 20 drug candidates on clinical trials. So the marine natural products has a very good potential for becoming the for becoming drugs for the drug discovery and to treat the human disease. Okay. And so this material basis of the marine drugs are those we call marine natural products. They are derived from the mollusks, sponges, soft corals, algae, mangroves, microbes, and so on. So with very different structural types. Okay, so they mainly contain like terpenoids, alkaloids, steroids, polypeptides, peptides, and many of the compounds comprise sulfur, phosphorus, and halogens like chlorine or bromine. So these are marine natural products, the base of the marine drugs. Okay, and so before going to my own science, I would like to do a little bit scientific education on how to do the marine drug discovery. Okay, so the process of the marine drug discovery. So with the marine biological material, okay, we would like to first do the extraction, isolation, and purification towards the secondary metabolites and do the structure illustration of the metabolites to know their structures. And then we will do the biological screening to find the bioactive metabolites. And then we will do the chemical synthesis, the structure modification, and the biological intensive biological study towards the drug leads. So this is the whole process from a marine biological material towards marine drug leads. And let's take the soft coral as an example. So first, we will cut it into pieces and then do the extraction and then the partition and evaporation to have the crude extract. So this is the extraction part. And then we will do the isolation with a lot of different chromatographies, such as silica gel, the LH20, to have the subfractions or the pure compounds. And then we will do the purification by a lot of instruments, such as the high performance liquid chromatography to have the pure compounds, the single compound from the marine soft corals. Okay, so with the single compound, next step is to know its structure. So we will do the structure determination by a lot of spectroscopic analysis, such as the UV, the infrared, to determine the fragments, and then the mass to know their molecular formula, and then the NMR to determine their planar structure. Okay, with the planar structure, next step is to do the stereochemistry determination, because every single molecule, they have their own chiral, their stereo centers sometimes. We will use like X-ray or X-ray diffraction analysis or circular dichroism study to determine their stereochemistry. And with the structure determined, the next step is to find their biological use. So we will do the high throughput screening or the activity-oriented screening, both in molecular level or in cell level. So in molecular level, like receptor to find their activity on receptor, ion channel, or kinase. And the cell level we will just try to find their cytotoxicity, their toxicity on the cancer cells and different other cells. Okay. And the next step is to do the chemical synthesis and structure modification to have the high activity while low toxicity molecules for potential drug leads. And then we will do the intensive biological activity test, both in vitro and in vivo, like in animal level. And then with these drug candidates, we will try to do the lead compound optimization and the target and mechanism confirmation to access to the market drugs. So by these two steps, we can have the drug candidates. And the next step, we will do the preclinical study, like the API technology, the pharmacokinetic and pharmacodynamic research, the safety research, to see if the candidates are toxic or not in the animal level, and then the dosage form to see if we can take it by table or other forms. And the last step is the clinic trial. Okay, so the clinic 1, clinic 2, clinic 3, clinic 4, and after the clinic trial, we can have the drugs on market. So this is the whole process of marine drug discovery. Okay, so with this process, there are by now there are 16 FDA approved marine drugs, and most of them, like more than 60 of the drugs come from the past 10 years, which means that marine drugs research are currently in a very booming stage. Okay, and by another side, we found that more than 70% of the marine drugs came from the marine invertebrates, especially the mollusks, which means that invertebrates are the main sources of marine drugs by now. Okay, and we think that mainly because the first, marine invertebrates are the most diverse species in the ocean, and the most studied invertebrates for medicinal use are like soft corals, sponges, or shellfish mollusks. So these animals have very how to say, characteristic symbols. You see, the mainly fixed growth or moving very slow. So they don't have obvious physical protection. They don't have shell to protect themselves. They can now move fast to avoid the prey of the other animals. So they can only produce the second metabolites to protect themselves, or to use them as chemical weapons. So these metabolites may have the potential for medicinal use. Okay, so that's why they are easier to find the medicinal compounds from the invertebrates. So there are also challenges for the marine natural product-based drug discovery. From this statistic, we can see that marine natural products discovery now in South China Sea, we can see also in China, we are like really in the booming stage. A lot of people, a lot of researchers go to the marine natural product discovery. And also from these two, we can see that sponges and corals are still the main sources from these two. And from this one, we can see that microorganisms derived marine natural products are rising. But there are already two main challenges for marine drug discovery. So the first one is how to efficiently obtain the diverse bioactive marine natural products. How to increase the chemical diversity, then we can increase the probability to find the marine drug leads. This is the first one. The second one is how to obtain the large amount of bioactive marine natural products, because we know that the marine resources are really precious. We cannot like collect a lot of the material to do the extraction. That is not eco-friendly. So we have to find other ways for the research. So with these two questions on mind, our research idea is just based on these two aspects. So the first one is how to improve the chemical diversity to efficiently obtain the bioactive marine natural products. So our strategy is to use the chemical ecology oriented discovery by two ecological clues. So the first one is the environmental stress, and the second one is prey-predator relationship, which I will go to the detail later. So with these strategies, we can more efficiently discover the new skeletons and highly active marine natural products. So with these highly active marine natural products, how to quickly and massively obtain these bioactive marine natural products for in-depth pharmacological research is another issue. And our strategy is to use the high efficient biomimetic synthesis and function or bioactivity driving synthesis. So the chemical synthesis to obtain the large amount of the compounds for pharmacological research, especially in the animal level, to accelerate the discovery of marine drug leads. Okay. So the first one, the chemical ecology for the two clues. I already told you that there are two clues. The first clue is the environmental stress. So this clue is the investigation, the formal investigation revealed that the different function secondary metabolites will be produced by the same species of marine organisms when they live in different living environments to adapt to these environmental change. So based on this clue, we have done a lot of chemical investigation of the soft corals, the same soft coral in different sea area. And we found that in different sea area, they can produce very diverse, different function secondary metabolites. So such as these different soft corals, we all find a lot of like molecules with the novel structures. These structures are never been found before. Okay, so and many of them have a lot of immunopotentiating activity or immunosuppressive activity and anti-inflammatory activities. Okay, so these are the beautiful structures that we have found from the marine sources. Okay, and the next clue is the prey-predator relationship. So this relationship based on that the investigation revealed that the mollusks can ingest the useful metabolites from their prey and convert them into their own functional molecules to serve as chemical weapons or chemical defensive compounds, so which enable them to have the potential for medicinal use. Okay, so such as the mollusk will take the sponge, corals, or algae as their prey. So our study, we have studied this kind of mollusk, Girona funebris, and its prey, this blue sponge. We find that they have very characteristic common natural products. So these kind of isoquinoline or their dimers. So these compounds are very characteristic compounds in these two animals. Okay, and we found that five of them. So these 5 ones we not only find in this sponge but also find it in this mollusk. So this could be a symbol of their prey and predator relationship. And based on the investigation, so they might be the chemical defensive or chemical weapons of the mollusk. Okay, so then we did the biological activity test screening. We found that these compounds have very strong anti-tumor activity, and their target inhibitory activity. So this is the strategy for us to be efficiently find the bioactive marine natural products. And based on this strategy, we also did the study of two different mollusks, like these two mollusks and their pre-sponges. Okay, this sponge and this red sponge. And we also found that they have very common some common terpenoids. Okay, you see the white ones and the common ones, and for these two, the yellow ones and the green ones are their common ones. So they all have common structures, and these common ones are not existed in the other, like soft corals or other species. So they are their specific symbol metabolites. And interestingly is these terpenoids all contain the nitrogen groups. So they are common nitrogenous terpenoids from these prey and predators. And we also find that they have very strong antitumor activities. Okay, so this is the strategy for having the highly bioactive marine natural products. So by these two clues, we not only improve the ratio of discovering novel skeletons, novel marine natural products, but also improve the ratio of finding the bioactive marine natural products, such as these structures. Okay, and we also published some lot of papers in the journals of natural products. Okay, so here is a case study about this chemical ecology. So I have already told you that we have two clues: environmental stress and pre-predator relationship. So this case is we combine these two clues and to inspire for the rapid discovery of new skeleton, of both new skeleton and bioactive marine natural products in photosynthetic mollusk. This one, okay, this is a Sarcoglossum mollusk. Okay, so this mollusk is very interesting. They prey on the algae, and they will come from the stimulation of strong light. So what they can do is they can store the chloroplasts in their intestinal cells. You see, these are the chloroplasts in the animals. Okay, so they are famous as the kleptoplasty. Okay, so for this animal, we can just imagine that they living in the seashore. The seashore have very strong light. They don't have any shell protection, so they are also moving slow. So they have to confront the strong light, and they have to do the self defense by what? By eating the algae and store the chloroplasts therein. Okay, so they have very intriguing ecological significance, and maybe they will have some potential for medicinal use. So our question is, will the bioactive molecules be generated related to these ecological phenomena or not? So we collect these kind of mollusks in the seashore of the Ximao Island in Sanya in Hainan province of China. And we did a systematic chemical investigation of these mollusks, and we found that there are like nearly 60 characteristic pyranol, pyranol containing molecules. They are all contained in pyranol groups. Okay, and there are also 8 novel skeleton molecules being isolated from this one with these 4 different types. Okay, and these are the 4 different types of novel skeletons, which means no such structural skeletons has been ever been found before. And interestingly, we found that all these 4 kind of molecules, they are racemates, which means they have they are a pair of enantiomers. Okay, so their stereochemistry are just opposite. So which give a very big challenge for us to determine their stereochemistry. So we first did the chiral separation of these racemates, and then we used the NMR calculation, X-ray diffraction, and the ECD calculation, and so on for the determination of their relative and or absolute configuration. Okay. And next step, just want to compare the new skeletons with the pyranal co-isolates. So these are the pyranal co-isolates, and we found that they do have some common fragments, so which makes us to think about if they have some biosynthetic correlation or not. Okay, so we then did a literature investigation and did a proposal of their biosynthetic pathway, and from these pyranal precursor, we can these can do some photo-induced cyclization and acid-induced microaddition and so on to have these new skeletons. So we achieved the chemical communication from the pyranal precursors to the new skeletons. And next, we also did the biomimetic synthesis of the natural new skeleton 1. So the first one is the Meissner blue induced the Diels-Alder reaction of the oxygen and this diene, and then a Lewis acid promoted novel cascade cyclization towards this one. So this is a very efficient synthetic way to access to the marine natural products. Okay, and we also find a synthetic new skeleton in this condition, and then since this we did a biomimetic synthesis, so we are assuming that if the the synthetic new skeleton are originally from a natural product from the mollusk or not. So we did the fishing experiment. We did the LC mass analysis of the crude extract of the mollusk and this synthetic compound, and by making the calculation of the cosine similarity of mass, we make sure that compound 11 was a trace natural product in the mollusk. So this should be a natural product in the mollusk, while because it is a trace amount, so we did not isolate it, but we have proved by this mass study. Okay, so which give us a clue that the biomimetic synthesis can guide us for the discovery of more and more trace natural new skeletons in the in the nature. And in terms to the biological activity, so we have isolated several these kind of peroxy compounds. So we first have thought about by evaluating their anti-malarial activity, because we know that artemisinin have this kind of peroxy bridge. So we first screening this activity, and we find that not only these 4 ones, but also these 4 new skeletons can 100% inhibit the growth of the plasmodium in 10 milligram millimole the concentration, which means they are very high activity on the of the antimalarial. So finally, this research work we have been discovered not only new skeletons, but illustrated their catalytic biosynthetic pathway. We also revealed the molecular mechanism of the photosynthesis of the channels towards the new skeletons by the mollusk to confront the strong lights. Okay, and we also developed the biomimetic synthesis to guide the discovery of the tree's active compounds in nature, providing a new idea for the study of chemical diversity. So, so in the future, we can do the biomimetic synthesis to access to the novel skeleton. So this research work we have been able to publish in Angewandte Chem, so one of the famous chemistry journal, okay, as a research article. And we find discovery of so many bioactive molecules from the marine sources. So we are thinking how to obtain large amount of these bioactive compounds, since it is, you know, in order to eco-friendly, we cannot collect a lot of the marine materials for extraction. So for us, our strategy is to do the synthesis, okay, chemical synthesis, so biomimetic synthesis or function or activity driving synthesis. So biomimetic synthesis, as I already told you, for that biomimetic synthesis, the efficiency of the biomimetic synthesis is better than the traditional ones. So we are able to access to the synthesis, the bioactive marine natural products in a shorter time, and to access to large amount of them. And for the function or activity driving synthesis, that is some noise. Is it okay? Okay, sorry. So the function-oriented synthesis, our strategy is not to do the total synthesis of the complex molecules, because it is really time consuming and economic consuming. Okay, so we are inspired by the skeletons of the bioactive compounds, and to take the skeletons and to just want to shorten the synthetic roles and to simplify the synthetic strategies. Okay, and then we are able to quickly synthesize a lot, like hundreds of derivatives, and then we 10 of the strong bioactive compounds, we are able to do the gram scale synthesis. And finally, we found 3 drug leads, these 3 drug leads, which means we can accelerate the discovery of drug leads by this function or activity driven synthesis. Just want to show you some simple examples for the biological bioactivity driving synthesis, BDS. So by using this strategy, we can construct the natural product like compound library of the bioactive marine natural product for the deep function or bioactivity mining. Okay, so the first small example is, so the this mollusk derived the compounds Kristiana B. So this compound have a very novel skeleton with several different kind of bioactivities. So for us, we did the fragment analysis, and then we did the synthesis simplification to have a library of this marine natural product derivative, and then we did the high throughput screening, okay, to find one compound have the immunosuppressive activity. And then we did several rounds of the bioactivity driving synthesis to have this compound whose T-cell inhibitory activity have increased like 85 folds, okay. And the greatest advantage of this compound is low toxicity, and its selectivity index is superior to that of the positive control, the CSA. Okay, so then we did the mechanism study of the immune response of this compound on the T or B cell receptors, which enable to elucidate the mechanism of the activity of this compound. And with this research, we also published in the Mass Chemistry journal of Mass Chemistry. And with this successful example, so we then did a study of this molecule. This is a sponge-associated fungi derived polysaccharides. Okay, so it also have multi-bioactivities, but it has very high toxicity. So our strategy is to inspired by the structure and to do the modification, to do the biological activity driving synthesis. And we finally we access to this derivative, this compound. It has the in vitro and anti-inflammatory activity increased like 20-fold, and it also has neuroprotective effects, so which stimulated us to do, and it also has very low toxicity, so which stimulated us to do the in vivo study in the animal level. Okay, so we first did the in vivo anti-neuroinflammation improves the depressive-like behavior study. So the open field experiment, we find that by adding, so this is like the running rows of the mouse. So by the negative control, we see that the mouse is kind of depressive. They don't want to move. Okay, and then when we add our molecule, like when we add the 1 milligram per kilogram of the molecule, you see the mouse is willing to move, is moving much better than the control. And then when we add the more concentration of the molecules, the mouse is able to move very like ordinary, like the positive like the positive control. So which means it has a very good activity on the improve the depressive like behavior of the mouse. And then we also did the anti-cerebral ischemia injury, the study of our compound on the rat. Okay, and we found also when adding our compound, you see, so these are the ischemic model, with the white ones is the bad, with the blood cannot be go through. And so by adding our compound, you see it has very improved, very much better improved the situation of the injury. Okay, so which means our compound have very very strong both in vitro and in vivo activity. So which stimulated us to find the mechanism, to find we want to find the target of this molecule. So then we did the target research. We do the design and synthesis of the photo affinity probe, did the probe of the compound, and then we did the target fission, and then we did the target validation by several biological experiments, which I will not go to the details since I think it will be more difficult. Just want to tell you that by this kind of study, we are able to find that this kinase M2, PKM2, is a target of this compound for response to their activity. Okay, so finally, by this research, we are not only discovered the lead compound to treat the inflammation-related disease such as ischemia, stroke. We also find PKM2 at the first time to be a new target for neuroinflammation and related brain disease. Okay, we also illustrated the mechanism of this compound for anti-neuroinflammation by inhibiting the glycolysis through the PKM2 target protein. And finally, we provided a good research idea for the development of drugs for the treatment of ischemic stroke-related disease. So this research also published in Angwan as a research article, which is collaborated with another biological group in the China Pharmaceutical University. Okay. So so by this study, you see, we are able to get a marine drug list. We see that this is drug candidates. Okay, so by this stage, we are now in this stage. So from this stage to the market drugs, as I already told you, there are still 2 steps. So the first step is preclinical study. Okay, these four aspects, and the final step is the clinical trials. So that is the 2 steps that we still need to do towards the new marine drug. Okay. Okay, so at last, just a perspective. As we know that I already told you today, for the chemical ecology, we have the clue of environmental stress. We have the clue of prey and predator relationship, but also there is another relationship that we have to consider, which is symbiotic relationship. Okay, because during the previous studies, we found that a lot of bioactive molecules discovered from the marine animals, like marine invertebrates, like this Spanish dancer, this beautiful Spanish dancer, their analogs can be found in microbes. Okay, as we are now confronting the problems of the limited resources of the marine animals and the high cost and hard to collect of the marine animals or other marine resources. So we can study the symbiotic relationship, which we can just try to find their analogs of the active molecules from the microbes, and then we can either easy to culture the microbes, or we can do the fermentation or heterologous expression of the microbes to access to the large amount of the bioactive marine natural products towards new drug lists. Okay, so this would be a very interesting topic. And as I already saw from the video of the MERO Foundation, that you have a lot of very interesting marine samples, not only the invertebrates, nudibranchs, and a lot of marine microbes, bacteria, or other like fungi. So I really would like to, if we have the opportunity to collaborate to do the marine drug discovery. Okay, so the very last slides is just to go back to my topic, the blue ocean drug discovery. As we know that there are over 70% of the Earth's surface area is ocean, and the total marine biomass accounts for the 87% of the Earth. But however, the current research, our research, like I already told you, is like less than 1% of the investigated marine biological resources. So our research is only a tip of the iceberg, and more secrets of the ocean are waiting for us, waiting for you guys to explore. So that's my topic today. And last, I would like to thank. Okay, so first, I would like to thank Professor Xu-Wen. So he is like a big boss of our research. He guided us to especially bring me into the marine natural product discovery, this research area. And I also want to thank Yao Ligong for the sample collection. So he can dive with me into the sea to collect materials. And these 4 students for the case study that I already told you. And I also want to thank our collaborators like Kai Chen from the Marine Drug Open Studio, Kristiana from Eco Polytechnic, which is who is my PhD supervisor, and Li Jia, Tang Wei, Luo Chen from SIM, and Pang Tao from China Pharmaceutical University, Jiang Lubin from the Shanghai Pastoral Institute, and Wang Hong from the Zhejiang University of Technology. And also, I would want to thank the support of these foundations for my financial support, because we have to have funds to do so many researches. And also, last, thanks to the MERO Foundation for this kind of for this very good opportunity for me to communicate with you guys. Okay, thank you so much, guys. Thanks. If you want to discuss with me, my email are here. My emails are here. You can feel free to contact me. Okay.
Yes, thank you very much, Prof. Xu-Wen for very wonderful and interesting presentation. This is a very good knowledge and opportunity for us in Indonesia, because we need to make our product to become release and benefit to the society. So, Pak Wayan, would you like to help me to introduce more before the question and discussion, Prof. Xu-Wen? So we will have question and discussion. So please stay. Please, Pak Wayan.
Okay. All right. Can you hear my voice, Rhesi?
Yes, very clear.
Okay. So once again, thank you very much, Prof. Xu-Wen, from your fantastic presentation. I was overwhelmed and I was thrilled to see your presentation. The way you present your slides are just fantastic, and the content of your presentation and the results are excellent. 4 thumbs up for you. Okay. All right.
But yeah, I would have, I would add a thank acknowledge to you, because without you I cannot have this opportunity. So I would like to thank you for this very wonderful invitation, and I would like to thank that we have met in Italy. Then we can have the opportunity to communicate with each other, and we have the opportunity to have this a lot of very good Indonesia audience for me.
Yes, you are welcome, sir. So when I was proposing you as the speaker for the webinar to the MERO team, the MERO teams expect that you are kind of a person who was in 50s or 60s, because you are a professor already. But then when I send the MERO team the pictures of you and the CV as well, they were surprised. You look like a K-pop artist or something like that. And then, but at your early, let's say at the beginning of your independent research, you have already such an impressive results, as you explained today to us. So they can see now who you are and your research actually. Okay, so please let me make 2 comments before allowing audience to ask questions. The first one, that you are starting your research from traditional, let's say a traditional approach in natural products, and then start from isolation and structure elucidation, and the most difficult part is stereochemistry configuration of the determination. So this is very difficult. And last, it's about synthetic approach, which is your strong background. And then we actually need expert like you in Indonesia. We need to train the next generation young scientists in Indonesia to study marine natural products, like exactly what you are doing in China. However, there is a traditional classical problems here we have facing now, where a lack of facilities in certain regions hamper our effort to do that. But we don't give up. Okay, so that's why we're expanding our network where we can have a possible collaboration. And you are already explaining to us that you are willing to do that. So it's very grateful opportunity for us. And now in Indonesia, my second comment is that there's a new era of research and education in Indonesia in the formation of BRIN. So this is kind of a National Research and Innovation Agency, BRIN. So as Indonesian people knows, this is a good opportunity for us because scientists and students and lecturers can have a mobility exchange from one university or to other research facilities, so we can sharing resources and also much more easier to do collaboration. So in this occasion, I would like to offer the audience, students, lecturers, scientists in this room to open up an opportunity to have a sharing, a joint research here, because it's new era in Indonesia in the BRIN era. The next new thing is in the era of MBKM or Campus Merdeka or independent campus for a glorious Indonesia. In this era, students from university now are welcome, and we have to facilitate them to study and to doing research and to do in-job training for more than 2 semesters or 1 year outside their university. So what we are providing here from MERO is that we have opened up an opportunity of facilities, welcome you to do MBKM here. And what we have in our network, such as Prof. Xu-Wen are also included in this networking. So we are expanding now the opportunities. So next is your time to contact us, to contact Prof. Xu-Wen directly if you have any ideas, questions, or something to comment about doing marine natural products in Indonesia. So this is a great opportunity. So Rhesi, I think that's all my comments about Prof. Xu-Wen. And now please let the audience to raise their question. And I believe from 80 more audience, there must be some questions they want to raise. Okay, so thank you very much.
Yes, thank you very much, Pak Wayan. So now it's time for question and answer for the discussion. So if you have any question, you can raise hand or you can put in the chat box, or if with Bahasa, it's also okay. I will read and deliver. So any question? Okay, while we are waiting, so I also note several questions that's already delivered and come to me. That's to Prof. Xu-Wen. This is about the chemical part, Prof. Xu-Wen. In the environment like Indonesia, we have several conditions of the sea, like clear environment and bad environment. So if we do sampling, like you said, that the environment very affect the chemical producing chemical compounds. So which one better, the clear environment or bad environment for the catch the targeting product of chemical, for example, for antibacterial, anti-malarial. So because we have so many spots to sampling the sample.
Okay.
Yes.
Okay, I got your question. Okay.
Yes.
So the thing is, so experimental stress, the clue what we want to use is to see every clue. We just want to find that the like soft-corals or mollusks, they can produce the like toxic. There are some environment for them to produce to stimulate them to produce the chemical weapons or chemical defensive compounds confront the environment. So it should not be the environment that make the animals to feel very comfortable. They don't have to. I mean, if the water is very beautiful and there are not a lot of predators, they don't have any threats. So this kind of environment may be not good for them to produce the toxic compounds, toxic molecules for them. So it should be the environment that maybe first the environment, the water is not so I mean no good nutrition, or there are a lot of interspecies like competition, a lot of competition between them, which make them could produce more like this kind of defensive or chemical weapons, this kind of molecules. These kind of molecules could be active compounds. Okay.
Okay. So very clear that
Probably.
probably.
Hands up.
Yeah. Yes. Thank you for the answer. So Rifki Sufian from Undip. So please, your time to ask.
Yes, thank you, Ms. Rhesi. Okay, I want to ask a question to Professor Xu-Wen. So my last research for my undergraduate thesis is about antibacterial activity from marine associated bacteria from Gorgonian. So when I was doing my research, there's some difficult for isolating the targeted compound. The thing is, when we do a preliminary antibacterial activity using agar plug method, we got a positive result. But after we want to extract the crude extract from bacteria using some solvent and something like that, and then we doing some antibacterial test again, we got a negative result. And I think in Universitas Diponegoro we still difficult to do it. Do you have any suggestion to maybe to make the result is positive? I think that's all my question. Thank you.
Okay, this is a very good question, in fact, and it's a very good. It is a difficulty that confronted by the natural product chemists. So what you said, it should be the bioactivity guided isolation, right? You want to test the antibacterial activity, so you test the antibacterial activity of the crude extract, and then when you do the isolation, okay, and you cannot find the antibacterial fraction, right?
Yes. First of all, we do a preliminary test using agar plug method. We just use the associated bacteria against the pathogen, and we got a clear zone. But when we do the next antibacterial test with a crude extract of its bacteria, we don't get a clear zone.
Yeah. So you did the antibacterial test with the crude extract of your samples, right? Of your materials.
Yes.
Or not? Yes.
Yeah.
Yes.
Using the crude extract.
And then when you do the isolation to make it to sub fractions, you cannot find the bioactive fractions, right?
Yes, there is no activity after we do a
extract. That's a problem. So that's why the biological activity guided isolation are not so often to to be proposed by the natural chemists, because they always will come from these kind of problems. Okay, so always they will lose the activity, because sometimes the crude structure, you see, there are like thousands components. You really don't know. Maybe it's not one single component to make the to respond for the bioactivity. Maybe it's a combination of several components to have this activity. And I think that is very similar to the traditional Chinese medicine. So we will combine a lot of herbs together to make that to treat the disease. But if we use want to find a single compound, we cannot find the single compound who is responsible for this one. So what I suggest of you, either you do the systematic chemical investigation of the structure to find as many single compounds as you can, and then do the test with the single compounds. With the single compounds, you will know really which one, and you should find this. I mean the how to say the large amount. I mean which compound is the most contains the most in this material. So you will start from these ones, and then the minor ones. And this is one option, is to isolate all the single compounds. And the other option is to how to say, to when you have the fractions, to test all the sub-fractions and to increase the concentrations of the fractions that you have to test, to increase it, because sometimes the compounds, because they are minor compounds in the fractions, so their concentrations may be not very high to have this activity. So the other way is to increase the concentration, and then if which one you have see the activity, then followed by this fraction to do the further isolation and purification. Okay. And you can if you have any other such questions or problems, you can just contact me. We can communicate by email or other, because now I don't know really the details of the problems. Maybe we can communicate later on by email if you want. Okay.
Okay, thank you, Professor, for your suggestion. And maybe I want to ask some question again, but not related to my first question. Is it okay?
Yes,
Yes,
definitely.
Okay, so Professor, have you ever researched about biochemical compound from marine worms, some marine worms? Because in my hometown in Lombok Island, there is some tradition of these people in the coastal area. They eat the worm, the marine worm, and they believe that there are some medical
sense.
effect from medical effect that given by eating the marine worms. I think it's about for a diabetic or something, or some disease.
Really? That's great. That's interesting. I don't know what kind of marine worms are you talking about, because maybe maybe there is a scientific name of this marine worm. So I didn't do it, but I'm very willing to do such, like it's just kind of like a traditional Chinese medicine, like we are using it by local people to treat some disease, but there's no scientific investigation or chemical or biological investigation of this one. So I would really very like, I don't know, maybe in China we also have this kind of marine worm, right? I don't know which kind of marine worm, so contact me if you want. Tell me which kind of marine worm, if I can find it in China, Or if not, then maybe we can collaborate with MERO Foundation to do this collaboration to collect this marine worm and then do the pharmaceutical study. Okay, very good. Thanks.
Okay, thank you, Professor, for your answer. I think that's all for me. Thank you.
Thanks.
Thank you, Rifki.
Rifki.
Any question? Any more question from the participant?
Okay, seems like they are quiet, but yeah, in
I
the
think
chat room it's also empty. Okay, Rhesi?
Yes,
I think
Pak. Yes, Pak Wayan, you would like to asking?
Oh yes, if no one asking question yet, then I have one question to Prof. Xu-Wen.
Yes,
Yeah.
please.
So in MERO Foundation, we are now growing about 200 bacteria strains isolated from associate bacteria from sponges, Ascidian, and so on. So it's almost close to 200 since the last 6 months. And we are doing screening with anti-malaria and antibacteria. So next, our next challenge is how to screen the NMR, the chemistry of the extract. This is the next challenge. Yes, we are dying for the NMR actually. But anyway, that's not my question right now. So my question is that, do you have any certain experience or strategy or approach how to deal with such a big number of extracts, then to come up with few hits that you could have at the end of the day, something like a lead compounds, as you explained. So what is the best approach? Shall we use a bioassay guided isolation or NMR guided isolation, or what is your suggestion about this?
Well, as I told you, for us, our idea, you see, we use chemical ecology. So like pre-predator relationship, how to just to explain, you see, because if we can find the common compounds from these two species, then it should have some. So this should be like ingest from one by one animal to from another animal. So this kind of metabolites should be have some like ecological or functions or biological functions. So which has a potential to be used by for the how to say, for the medicinal use. So by this one, we are just like first we are selecting the materials. So for by example, we want to select the mollusks which eating the special sponge, or we want to collect the soft corals living in the very harsh environments, and then we do the isolation. And for the isolation process, to be honest, I think the best way is to isolate and purify as many compounds as you can, because by now the activity-based the isolation or NMR-based isolation or don't show the very positive results. But another one I think is good for thinking is molecular networking, the mass guided discovery, which is better for us to quickly identify the known compounds and the new compounds. So by the cruise extract, we will do the LC mass analysis, and then to pull the results to the library, and then they will generate a networking to see which type of compounds are together, and this type of compounds are there new ones or not. If there are new ones, we will focus on this fraction to do the isolation. So this may be an option for finding finding new compounds. But for the bioactive compounds, to be honest, it has to be based on the first the previous study about the compounds. If we already have this kind of like skeletons could have the bioactivity, or just to do the high throughput screening of the compounds. Yes, for us, I think is the way to find.
Okay. Yes, perfect. I do agree with your comments that for the future, the replication such a big a lot of extracts, GNPS is the solution, one of the solutions, I think, where GNPS is the global natural product social molecular networking. So it's just by having the LC-MS data, then we can have insight which extracts that contains possibly new molecules. But in order to do that, LC-MS is the must-have instrument, which we don't have now here, and it's quite expensive.
Okay.
So, all right. And then, but yeah, GNPS is good, it's a future. So that's why we are aiming to have the next our MERO webinar would be about GNPS. So we need to find someone who who really understand, and then professional, and then who can share their knowledge how to carry out. And of course, and even we are planning to have not an online but an offline workshop, the training about GNPS, which must be quite beneficial for us. Okay. Anyway, thank you for your comments. Yes, Rhesi, thank you.
Yes, thank you, Pak Wayan. There is a question from YouTube from Dian Sari Maisaro. So I will read this. Prof. Xu-Wen, I have the result of NMR spectro, but I have difficult step for structure elucidation from my sample. The sample is single compound from sponge. Maybe any tips how to solve it? Thank you.
What is last sentence?
Yes, the last is the sample is single compound from sponge. Maybe any tips how to solve it? How to solve
how to illustrate the structure, right? That would be a very long time course if I will teach you how to do the illustration. The first, if you have the proton NMR, then maybe you can just know what kind of molecule it could be, and then you have to have a carbon NMR. I don't know if he has all the NMR structure or not. If he has the carbon NMR, then we can already determine if it is known compounds or new compounds. If it is known compounds, we just put the carbon NMR data into the database and then click search. If we can search one, then it is no one, then okay, down. If there are like 70 similarity or 80 similarity, then probably it is new compounds, but it's the analog of the known compounds. So it will be easy to use the other like two-dimensional NMR spectrum analysis. And if it is you cannot find any similarity, then perfect. That would be a novel skeleton. And you will do the whole two-dimensional spectroscopic analysis, and then to access the planar structure, as I told you, then the stereochemistry. Okay, so if he want to know, first, I think he can search for Wayan. Wayan should be an expert for this structure determination. And then if you have any question, you can also send me this the spectrum to analyze, no problem. tell him in YouTube.
Yes. Yes, thank you, Prof. Xu-Wen. This is the last question from chat as well. This is quite different question, but this is about COVID. So, do you know what is the product from marine natural product that maybe now they are in developing to how to make the immune stronger to attack the coronavirus? Is there any product from marine?
Against the virus, the coronavirus, right? To find the antivirus marine natural products, right? In fact, you know, there is already a drug which is on market in China as Voodoo. You know, this is kind of how to say how to say the structure of these compounds is already like 3 marine sponge derived drugs. So in fact, we can do is to use the already reported marine antivirus drug to screen the coronavirus to see if there are activity or not. And for us, we are doing this kind of research now. We first do the do the structure modification of the currently antivirus, anti-HIV or anti-HPV drugs, marine drugs, to do the structure modification, and then we want to test the coronavirus to see if they have any activity or not. I think it's it should be the easiest way, or you should take shorter time, because if you want to find a new compounds, it will take you like 15 to 20 years to develop a drug. But if you derive it from already the market drug, it will be more easier. It will be easier and takes shorter time. Okay, thanks.
Yes. Okay. Thank you for the clear answer. So we already the time. I think Prof. Xu-Wen already 30-minute discussion. So this is very nice discussion, and of course we would like to discuss more and then doing research together and collaboration. So next time MERO Foundation will contact you. Okay, so for the next session, we will go to closing, but before that, we will do documentation together, and then we will give the certificate to you as our appreciation for your time. So yes, so I think this is still share screen, yeah, Prof. Xu-Wen.
I want you to
yes. Okay. So I give to the committee for documentation. Dila or Ayu or Risma. We go to documentation first. So for the participant, please open your cam.
Wow.
Okay. Masih on mute, Dila. Yes.
Okay. Let me take a screenshot first. And 3, 2, 1. Okay. In second page, please. 3, 2, 1.
Thank you. All day.
Okay, thank you very much, Dila. And the next session, we will give the certificates as a symbolis. Please share the certificate. Okay, please take picture also. So we are so thank you very much, Prof. Xu-Wen for your time and knowledge to all of us in this webinar, Bioprospecting of Marine Natural Product 2022.
Thank you so much.
Yes, yes. So we will deliver by email. And then we end to the session for today, Prof. Xu-Wen and so we close. We thank you very much, and also for all of participants that still here until the last session from everywhere. I know you already learn many things. So we also waiting you for the next webinar. In here, we also see several participants that already joined in the previous webinar. So we really appreciate for your time, all of you, Prof. Xu-Wen, and also special thanks for the committee that prepare for this webinar. Yeah, so
Okay, thank you.
I close and thank you very much. Pak
Thank you,
Wayan.
everyone. Thank
Okay,
you,
thank
everyone.
you. Bye.
So, yeah, okay, bye. Looking forward to meeting you somewhere in Shanghai in the future.
Yes, I hope so. Okay. Welcome to Shanghai. Welcome to China.
Yes, yes.
Welcome
Thank
you
you,
guys.
thank
All
you.
you guys.
Okay.
And
okay,
we are
see ya.
waiting to MERO Foundation.
I also would like to go to Indonesia to dive in the very beautiful water of Indonesia.
Yes, please.
Yes, we are waiting you, Prof. Xu-Wen.
Okay, bye bye.
In
Ciao.
Okay,
Bye bye.
bye-bye.
Bye.
Bye-bye.
Thank you.
Thank you very much.